Okay, so we'll get started. It's going to be a short class, but I wanted to introduce you to this interesting topic of genome-wide association studies. Um, any idea about what these studies could entail? What 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 is the general goal? What is genome-wide? What is association studies? What are SNPs? Has anyone heard of SNPs? SNPs? Single nucleotide polymorphism. Okay. Okay. So a SNP is a single nucleotide polymorphism, which is basically a position in a genome which can allow where some individuals can have one nucleotide, other individuals can have some other nucleotide, right? This can be both in the protein coding context. It could also be in the non-coding context, which means that it can either code for a protein or it can be in a region which does not necessarily code for a protein. So it is single nucleotide because it's at one position, so it's single and it's at the nucleotide, so that's why nucleotide. And then polymorphism because it basically takes two kind of shapes, which is it can theoretically allow 4 different nucleotides to happen at a particular location. And these are estimated, so this is very common, right? So your and my DNA is 99.9% similar, and 0.1% of these are basically SNPs, where you have at certain positions A and I might have a G and so on, or you have a C and I have a D, and everyone in this class can have a slightly different configuration of what exactly exists there. So theoretically, each SNP can have 4 alleles, but most SNPs will exist as 2 alleles, which means that if it is A, it will be G, or if it is A, it can be C, but not all sorts of permutation combination, and we'll see exactly in a minute why. How do they originate? So they originate because we all kind of like think of the most earliest humans. They must have had one particular copy of the DNA that everybody started to reproduce with, and as they reproduced and as we sort of age, there are 2 things that happen. First of all, as we age, the DNA will undergo mutation because the repair machinery is not able to keep up to everything. It tries to minimize the errors, but just because of there is some fidelity there, it will end up incorporating some error. And if this error happens to be in your reproductive cells, then the father or the mother can pass the mutation to their children. And if this keeps on happening again and again, if it doesn't get fixed, this particular nucleotide variant will become established in the population, right? So for example, the reason we are genetically all Indians have some shared characteristics. These characteristics arise basically based on the idea that our ancestors had some geographical separation from the rest of the world, and because of that they reproduced in a particular manner, right? So there was not a lot of mixing going on, let's say, with the American population, for example. And if that is actually true, then we will see a lot of similarities just because of our reproduction is becoming that is sort of playing a role in terms of which SNPs get passed on to generation. And eventually, these mutations, sometimes they're not deleterious, right? They're in the sense that they're not going to cause any particular damage or disease. They just keep getting passed on to generation. However, they can be functional in nature. For example, we have a particular kind of skin tone which could be associated with a particular SNP that has been passed on to us from multiple different generations. We have a particular eye color which runs in our family or which runs in overall Indian population. There's not a lot of blue-eyed people, for example, here. That's particularly because we don't have that SNP for causing the blue eye, for example, which is very common, for example, in European or American population, right? So the SNPs keep on getting passed on from one from one generation to the other, and if this keeps on happening, then eventually they will get fixed. So how do you think we can understand, or how can we profile SNPs? How can we understand where are these SNPs located? What if you were to do an experiment? How would you do it? How would you profile? This is mutations in general. How would you profile mutations generally? Like, We did an example in the class. That's one way to do it. How did we study mutations? Any example of mutation that you had studied in this class so far? Okay, gene knockouts. Okay, that is. Okay, sure. So they can do a mutant. You can mutate. You can do a CRISPR mutation there, and that's one way to do it. Any particular disease that comes into mind which had a mutation involved? Yeah, so sickle cell had one particular mutation involved. Right? So if you were to do an experiment to profile whether, let's say, I have sickle cell or not, how would you do it? Everything is known about sickle cell, where it happens in the HBB gene. HBB gene is located on chromosome 11. It happens at a particular location, location number 6. How would you do the experiment to profile whether I have sickle cell or not? Any theoretical experiment? Okay, so you'll align the normal gene with. Okay, so you will sequence my entire genome, or okay, then you will just basically sequence that one gene, right? So yeah, so rather than, if you know something about a particular mutation, if you have a lot of information about it already, like, so a lot of these SNPs, these are known to occur in and they are known to be causing something, and or not causing, but something associated with something, and we'll see what that association is in just a bit. But if you have prior information about these SNPs, then you can do a smarter experiment. You don't need to profile like the entire DNA sequence. You can do a targeted capture. You can just go to that particular gene or that particular location and then do this experiment, right? So this is exactly what is done in SNP arrays, right? So have you heard of something called 23andMe? No? 23andMe? You have heard. Yeah, so it's to find your ancestry. It's very, very common. Like, a lot of people do it in the US because they're looking for answers about like where did their ancestors came from, because mostly in America your family must have migrated at some point. Very few native people left there overall, right? It's not entirely true for Indians, because we have an ancestral North Indian and ancestral South Indian population, but like overall, in the first or the second generation, most of your ancestors would have been in India, would have already been in India, whereas that's not true for for Americans. So it's an assay which is used a lot to test for your ancestry. Like, what is the general idea of how exactly it works? It works with this idea that if you know where exactly the SNP is going to occur, you can design an oligonucleotide around it, right? So this oligonucleotide is nothing but it's a 50 nucleotide single-stranded DNA. You take this single-stranded DNA, and just because what we have continuously seen throughout the course, that things will hybridize, right? So if you take a DNA target, this is your DNA target, where you know that this particular DNA target harbors this particular SNP. You can design an oligonucleotide around it, and then you can perform some sort of a hybridization reaction and capture whether the hybridization happens or not. So the hybridization will happen irrespective of whether the base matching pair is exact or not. So for example, if I had a SNP here, which is C, it will still hybridize to it, but it will not happen with the highest efficiency. So it will prevent, in this case, it will prevent hybridization with the highest efficiency. If this was G and my oligonucleotide had a C here, this will hybridize much faster or at a much higher rate. Whereas this hybridization will be at a much slower or it won't happen in some way, right? But generally things are not as exact. This will still hybridize, but it will not happen with the same efficiency. And what these DNA hybrid arrays or DNA chip or all these assays, which I'm going to talk about in just a bit, tries to do is it tries to understand whether the hybridization is happening or not. So you can use, this is for example a GWAS slide which has each dot here is one kind of an oligonucleotide. So this is a GWAS slide with oligonucleotide tethered to it. And what you're essentially measuring is if the hybridization happens, it emits a signal. In this case, it's a red signal. If it happens to be emitting a signal, then that particular SNP is whatever is there in your oligonucleotide. If the hybridization does not happen, means you have a particular kind of a SNP. So you can use this information to figure out which parts of your DNA has a particular SNP entangled into it, right? It's not as specific, like this overall is just telling you whether there is a SNP or not. We want to be a bit more specific, and we'll see this in just a bit of exactly how do you figure out whether it's a C, it's a G or a T, okay? But the overall idea of how these arrays work is you do some sort of hybridization and you measure whether the hybridization is happening with high efficiency or not, okay? Is that general idea clear? Okay, right? So genetic testing is becoming very accessible in a way, and by accessible, I don't mean to do it to tell you that you should do it, but it's affordable. It's becoming affordable. For example, if you want to do your 23andMe, which it's a kit which looks like this. This is primarily for the US, where you can get your health and your ancestry figured out. What you have to do is very similar what we do in India. So there's an Indian company also which tries to do it. And again, none of this is, I'm not trying to endorse one company or the other company over anything. I'm just giving you examples of what exists in the market. It is called Map My Genome. What it does is it sends you a sample collection kit, you spit into it, you send out this sample, they will perform an analysis which will look like very similar to this analysis here, and they will tell you what kind of what kind of your genetic DNA, or what kind of DNA is given your SNPs, what kind of diseases are you probably predisposed to, or what is your ancestry. So again, the ancestry is not very much of importance in India because we kind of already know where we came from, because again, your first and second generation must have been in India, most of you. But in US, this is a very common thing to figure out where exactly your ancestors came from. And there are, if you go and read up about it, like you'll come across very interesting case studies where people have done this test just before they were getting married to realize that they were going to get married to their half brothers or half sisters, right? Which is something which you would not want to do generally. Yeah, it's an interesting kit, right? So there are like a lot of very interesting stories that 23andMe has revealed. Sometimes people, yeah, so go and read about it, like very interesting stories, right? So I did my 23andMe back in 2015, and this is the result I got. So nothing very interesting about it, right? So it claims that I am Central Asian, I am North Indian, and Pakistani, right? So there is no way to distinguish between at least that time there was no way to distinguish between all these three kind of categories. I think now you can be a bit more sure, and that's what it shows you here, that 96% I'm a North Indian and Pakistani. In fact, it gets it actually gets my state very correct. I actually come from Bihar. I have some bleed through of Bengali sort of genes in a way, and I'm broadly North Indian for all practical reasons, right? So I did this test was very excited when I did this. But when I got the reports, I was like, there's nothing that I've actually learned that I did not know, right? Like all of this is very common information. So that's one part of it, but there are interesting things that can actually come out of these SNP arrays, particularly from a disease point of view. You can understand which drugs will act on you or not, because not all drugs will behave in the same way given your genetic profile, right? So a lot of its application is towards figuring out what is the right therapy for you. That is number one sort of application. The second application is just figuring out, given your genetic makeup, which diseases are you predisposed to. So given I do this test, what is my probability of developing a diabetes 5 years or 10 years down the line? What is my probability of getting stunted or not having height which is above 5 feet or so on? Like there are all sorts of crazy characteristics that can be measured, and we will see in just a bit how exactly that is done. Okay, so these technologies broadly use either of these two companies, which is Affymetrix or Illumina. So Affymetrix arrays, the goal is to capture which SNP you have, right? So a particular SNP, for example, at a particular location could be A or a C. What you will do is you will design oligonucleotides specific to known locations where SNPs can occur. So you take the DNA sequences where you know that a particular SNP is known to cause something, right? And the way this is made possible is because number of studies would have organized or would have carried out previously that this particular mutation at this particular site causes, let's say, or is associated with, let's say, breast cancer, or this particular mutation in chromosome 11 causes sickle cell disease, or this particular mutation in chromosome 6 causes colon cancer, right? So this information has been published in literature, not primarily because of use of these assays, but in general, like in terms of, because people keep studying these diseases or these mutations overall. So if you have access to that information, what these companies do is they go back and they synthesize oligonucleotides which are catered to some specific locations. And broadly what you're trying to do is you're trying to do a hybridization, trying to capture whether a particular SNP emits a particular kind of a signal or not. And the way this is done is slightly different between Affymetrix and Illumina, but the general principle is very, very similar. So in Affymetrix, you have 25 mer probes. These are attached to your, again, sort of a glass bead or a glass slide. Your DNA will, so these are your probes which are attached. Now, if you take this particular DNA sequence, it will hybridize, even though there is a mutation here. So for example, if I can zoom in, I cannot zoom in unfortunately, but maybe you can see it, right? So this particular sequence is, so your probe here is ATC, this probe is AGC, TCT, CGT, right? So these are different sort of possibilities given this particular DNA location. What are the 25 mers that you can synthesize? Now if I take this probe ATC, I'll try to look where exactly the hybridization will happen, right? So ATC seems like it will hybridize here, right? So this, that's the actual sequence. It will hybridize based on the complementary sequence. So that will be TAG. So TAG happens to be not shown here, but you can imagine that there is a location where TAG happens and it will sort of cross hybridize. The second probe that you have here is AGC. So AGC is going to hybridize to again so TAG is a sequence that should happen here. Hold on. Okay, so these are coming actually from locations which are not part of your this sequence. What you will end up getting is there will be some hybridization that happens, which is not specific. Actually, that is the point of this figure. Is if this hybridization is exact, like wherever it is coming from, it is not coming from any bases that are shown here. But if the complementarity is very, very strong, this dimer or this sort of bead that you have will light up much strongly as compared to the bead that is shown here. Similarly here, which is this sequence now, AGA, this is going to hybridize in an exact fashion because you have A complementary to T, G to C, A to T again. So this will emit a much stronger signal. Whereas if you have the other allele, right? So if you have, or this is again a different allele where you have AGA, but the bead that you had was GCT, then this is not going to be an exact hybridization, so it will end up emitting a less signal. What you essentially end up quantifying, and this is shown for the remaining, for the other allele, C. So this AGA is replaced by CGA and so on. What this shows you is whichever allele you have, that particular probe will end up lighting up and you will know which particular SNP is present in your sample sequence. This is what Affymetrix does. In case of Illumina, the case is slightly different. You have again a bead which is attached to a different sequence. This is a 50 mer sequence, so it's slightly more specific as compared to Affymetrix. Now you have different probes which have slightly different colors. So you have A, C, T and G, A and T being red, C and G being green. As the extension happens, as you take your sequence and the polymerase tries to extend it, based on which particular sequence gets incorporated, it will emit a particular kind of signal. So for example, if I started from the C sequence, I will end up incorporating a G, so it will emit a green signal. But if I started from an A signal, it will end up incorporating a T here, and that will emit a red signal, and that can be quantified. So these are sort of the two broad differences between Affymetrix and Illumina. So Illumina is using colored fluorophore, And Affy matrix is basically using the intensity differences to figure out which particular SNP is present in your sample of interest. Is that broad idea clear? So when you send out your sequencing sample for sequencing or on your for your 23andMe or MapMy genome, this is what they do at the back end. Yes. So incorporating T, right? So you have a mutation here, right? So it can be either A or it could be either C. Now you have a probe which is complementary to this, right? So if you have a probe complementary to this, in this case there is only hybridization happening, whereas here it works by doing a single base extension, right? So in each point you will have at each point there will be some partial hybridization, so it doesn't have exact it doesn't have an exact complementary sequence to it, but it only has the primers here. So with these primers, you can have a DNA polymerase sort of start to do the extension. And when you do the extension, when you encounter this particular sequence here, right, so it will end up emitting a red signal. So this is very similar to what we do in case of actual sequencing. It's just that it is happening in case of Illumina on a bead, and these are like 50 mer sequences which you begin sort of cross hybridization with. Okay, so in this case, you are slightly more specific. You start with a 50 mer, right, and this sequence is overall complementary to it, but with the exceptions being that at certain sites where you know the SNPs can occur, those positions are, these K-mers are located adjacent to it, right? So it doesn't complementary, it doesn't do an exact complementation, so you have to do one round of extension. When you do that one round of extension, you will figure out whether is a T being incorporated or is a G being incorporated at this extension end, right? So you can think of this K-mer coming from this particular location or this particular location. Once it hybridizes, so this is like a 50 mer, and let's say this is 51st position. You have an overhang, and that overhang needs to be extended. Whatever gets sequenced, that will tell you whether it's a K-mer, it's a A or a C or a T or a G sequence. Okay. Okay, so we have a technology to associate, to sort of sequence or to figure out what SNPs are existing. Now we have to figure out if we can do this at scale, right? So here there's a chicken and egg problem, right? We can go to certain locations and we can do this because we know that those locations are known to be SNPs or known to be prone to be SNPs, because SNPs will not occur everywhere, right? Like in the sense that if you want to make a certain kind of a protein, one particular change can, if it disrupts that protein, most likely that SNP is not going to survive, right? So some of these SNPs are going to be more specifically in regions where you can profile them, where they're present in every other individual or like a particular population. But most of your other protein coding sequences will more or less be consistent, right? So the chicken and egg problem is as follows, that if you want to figure out what is the kind of function a particular SNP is doing, you need to do another experiment. So you have access to a technology which can tell you whether you have a SNP at a particular location or not. But at this point, you might not necessarily know what is the function that it is carrying out, right? So you have to figure out what are the phenotypes associated with a particular mutation. So how would you do it? You have access to a technology which can tell you where exactly your SNPs are located. You want to associate it with a phenotype, right? So that phenotype could be, is it associated with a disease, a cancer? Is it associated with type 2 diabetes, type 1 diabetes, obesity, longevity, like all sorts of possibilities, right? So which SNPs are associated with, for example, people who live longer? Is there a particular mutation that causes them to live longer? So how would you do this kind of experiment? You have access to technology which can tell you whether they have a particular mutation or not. So you'll find what? Sorry, can you go back? Okay, so you have mutation, but you're saying you should also get RNA-seq. Should. Okay, so let's assume that we don't want to do RNA-seq. Then how would you do it? That will go one level up, yes, that I agree. But if you were to not do it and you still want to, because right now the question that we are asking is, how does mutation change the phenotype? And in this case, the phenotype is a disease, right? It's a disease, or it's my height, or it's my body mass index, anything that you can think about. Color of the skin, color of the hair, blueness of your eyes, any particular trait that you can measure or observe can be a phenotype. I'm asking what mutations cause, for example, like if I have to sort of differentiate between, let's say, Europeans and Indians, what makes Europeans Europeans and Indians Indians? Like, how would you do that kind of experiment? Okay. Okay, so you'll have to go to the population level and then, okay, all sequence all, okay, and then next. Yeah, so very good, right? So that's very simple idea, right? Like, it's a very, you can think of this in the linear regression format, which is, let's say I collect people from different ancestry, Europeans, Africans, Indians, and let's say Han Chinese, right? So these are like 4 different subpopulations. I want to understand what is different between them. One way to do that is I collect like multiple individuals which are known to have that particular ancestry, and then figure out, if I have access to a technology like this, figure out how many, at which locations, which SNPs they have present, right? And then what you will end up seeing is people from one particular ancestry will have one particular kind of mutation overrepresented. It will come over again and again, again and again, again and again. And that is the general idea about genome-wide association studies, right? So this is called GWAS, or which is short form for genome-wide association studies. The idea is very simple. You collect data from multiple individuals. These individuals will have different traits. They can be coming from different cohorts. They could have different covariates. They could be different sexes, could have different BMIs, could have different eye colors, skin color, whatever, different ancestry. Then you do a genotyping, right? So what is genotyping? Genotyping is basically what we are doing here, is you're trying to profile what is the genetic makeup at a particular location for all of these individuals, and you're doing this at multiple locations. Rather than explicitly performing DNA sequencing, which would be more exact, what you're doing is you're taking these arrays and you're profiling it at, let's say, 100,000 or 500,000 locations, so that you know at those locations what is the genetic component of it. Is this individual always have an A at location 1? Does this individual have always C at location 1, and so on? Okay, and this is what is represented here. So for example, at one particular location, person 1 has G, the second person also has G, person 3 has, and 4 have CC, right? This has TTAA, this has GC, right? So the goal here is to figure out at each location what is their genetic makeup. Now once you have done this, the second step is very simple. It's just literally carrying out some sort of a linear regression or a logistic regression, which we partly covered in the class. But the idea is, given a particular trait that you can quantify, what is the association between this SNP, location 1 being GG, corresponding to the value that you're quantifying for that particular phenotype, right? So for example, this person, let's say if these two have GG, both of them have GG, maybe both of them have higher height. Whereas if they have CC, then both of them have lower height. What this will essentially correspond to is having a G means having a higher height. That is where the term association comes from. You cannot make it causal, but you're saying that having a G is associated with them having longer height, okay? Or similarly, you can do this for any kind of a trait. This is what is usually shown, and we'll see what exactly this particular kind of plot is called. So before actually we see what kind of this plot is called, just overall, what are the steps involved in a genome-wide association studies? You will collect patients or people from different cohorts, or this could be like if you're doing, let's say for example, in an Indian population, you want to figure out what is that disease what is that association of which SNP to type 2 diabetes. You will collect all sorts of people from different parts of India, perform their genotyping, so perform sort of a 23andMe for all possible locations, do some sort of quality control, then catalog at each location what is their genetic makeup. This sometimes you have to what is called imputation, which is that because these arrays are going to be noisy, you cannot actually figure out whether this person really has a G or a C. But if you know their ancestry, you can pool some information and do some sort of imputation. So we'll not focus exactly like too much on imputation part, but this is just to highlight that if you even did not profile that this particular person had a C, you can do some sort of clustering to figure out what is the most likely SNP that this particular person will have, maybe because it is very close to person 2. Okay? And after you've done that, you will perform an association testing to figure out how much of does the variation here explain the phenotypic trait. So in this case, the diabetes case, you will try to run a logistic regression to figure out, given that this SNP location has a frequency of 0.5 for G and C, how much of it explains the differences between whether those people have type 2 diabetes or not, right? So let's say if all the 4 people are going to be type 2 diabetic and have type 2 diabetes, this is not an informative SNP because all 4 of them have the same traits, right? So I don't really learn anything in the experiment. But let's say if these 2 persons have type 2 diabetes and these 2 do not have it, then I will say that this SNP is associated with type 2 diabetes, whereas having this SNP means that you're going to be free of diabetes, right? You can also, if you do this kind of study, let's say I do it in India, somebody goes to China and repeats the same study, there is no guarantee that whatever I find here will be found exactly in the Chinese population as well, right? So we'll see this in a bit of, this is a problem of this entire field of polygenic risk scoring. So most of these diseases that we talk about generally are not caused by one disease. Like these are, they're not caused by one gene. Most of the diseases that we see usually are caused by multiple different diseases. So there are number of genes associated with each disease. At least most of the diseases that we don't understand, including cancer, is because it's not caused by one particular gene, it's caused by multiple genes. So what I can do is, if I do this in India and then I go and do it in China, I can do a meta-analysis. I would have come up with these associations in Indian population, I could have come up with these populations in Chinese population. Then I could find out like which of these SNPs reproduce in terms of their replicability, right? And then maybe perform a final experiment in an actual population where I say that, okay, if I know that if I go to a lab, for example, and do a particular kind of a mutation, then does that lead to that particular mouse, for example, getting a kind of a diabetes, right? So this is at this point very much in the form of association. It requires few more steps to perform to gain causality. You cannot associate causality with just this analysis. Okay, so how do you test for an association, right? So this is very, very simple if you think about it. You can have, for example, the example that we saw previously, I could encode this as 0, 1 and 2. For example, getting a G could be 0, getting a C would be 1, and getting a T could be 2. It could be anything, arbitrary chosen values, does not matter. I could get that is my x-axis here, and on the y-axis I'm plotting a phenotypic score. So this phenotypic score could be anything. It could be the severity of disease, it could be a body mass index, it could be your blood glucose levels, whatever you want. Like this is a phenotype that you define. All you're doing basically is you're trying to run a regression, and slightly fancy regression, which we'll not get into. But broadly, the goal is to test whether does the variation in X help you explain the variation that you see in Y. The variation X being the variation in your genotype, the variation in Y being the variation in your phenotype. And if you do this kind of test, for each location in the chromosome that you're testing, each SNP that you're testing, you calculate a p-value. That p-value is a measure of, in a way, the measure of the strength of association. So for example, if I see such a strong line like this, I will say that there is a very strong association of this genotype to the phenotype that you're observing. Okay. So this plot that you're seeing here, the x-axis is the chromosome location of the SNP. Each dot here is, so these are chromosomes, and within those boundaries, what is the actual location of the SNP? And on the y-axis, we're plotting the observed p-value, right? So the p-value here, in a way, quantifies the strength of association of that SNP to that particular phenotype. So the larger, this is minus log 10 p-value, because p-value, the smaller it is, the better the more confidence you have. So the way to quantify this is to take log 10 and then take a negative sign. Why should I do this? Why minus log 10 p? Why not just p-value? So the x-axis here is chromosome location of the SNP within that chromosome, so 1 to 23. Each dot here, like whatever you're seeing, which you're not able to see here, is one particular location in that particular chromosome, okay? The y-axis is the observed minus log 10 p-value, which measures the strength of association of, or the sort of the slope of this line in a way. Why should I plot minus log 10 p-value? Why not just p-value? Yes, very, very right? So the p-value is going to be very, very small. If your strength of association is very large, these will be right. For example, honestly, at this point, this is a very crazy number, like 100, which minus log 10 p, 100 means 10 to the power minus 100, which I believe it's, I think, smaller than the number of molecules on this earth, I think, right? So it's 10 to the power 52 or something. Anyway, but this is very common, like in terms of the values that you see here. I might not be right about the number of molecules, but I think 10 to the power 100 is still very, very, but this is very common in this field. So what this plot is showing you is this particular SNP is associated, this particular location on somewhere on chromosome 19, you have a very high chance of this, whatever mutation you have here, to be associated to your phenotype, which could be again anything. So these are called Manhattan plots. Any idea why these are called Manhattan plots? Yeah, it's the Manhattan skyline. So Manhattan is New York City for all practical reasons, but Manhattan is one of the suburbs of New York City. A very, very beautiful place to be in. This somehow, like when the first plots started coming in, you actually go to any part of New York, not any part of New York, but most parts of New York, you'll be able to see this kind of structure where there are like very tall buildings followed by the old buildings which were like, smaller. Right? So that's why this is called a Manhattan plot. On the plot here, we are just plotting what is the expected value and the observed p-value. What this shows you is if there are SNPs that are associated, how many of them, what is sort of the degree of separation based on what you expect just because, of random chance, again, very loosely speaking, versus what you're observing their p-values to be. Okay. They can scale, this slide does not have a title, but what I wanted to show you here is the number of SNPs that you identify to be associated can be a function of the sample size, right? So the number of patients or the number of people you included in your cohort study could end up identifying more number of loci. So loci is the plural of locus. Locus is the location in the chromosome. What this particular plot is trying to show you is different traits. So this is BMI, this is height, and this is some sort of weight height adjusted BMI, okay? Doesn't matter exactly what this is. What this is trying to show you is different traits. So I can test the same population for different traits. I could have measured their BMI, I could have measured their height, but as my sample size increases, there's one thing that's happening, which is the number of locus or number of loci which are associated with BMI, my body mass index, increases. This is kind of natural to think about, because the more people you encounter, the more is the variation that you're sort of capturing, and that will give you more statistical power to detect things, right? So that's why the number of loci here increases. The second thing this plot is trying to show you is With the same set of people, the number of locus or loci that you identify for a particular trait can be different. While there is a lot of information available that the height in the genetic makeup which tells you height, BMI is harder to get associated with, or BMI in another way is harder to predict, right? So you can think of this as a way to understand, for example, how obesity is harder to characterize, though height is easier to characterize in a way. I can easily figure out which mutations causes, is associated with height, but if I have to do the same thing for BMI, which is sort of associated with obesity, that's much harder. That just tells you that this is just genetics, okay? This is complicated. Not everything will have enough power or enough things associated with it. Okay, so you have identified these variants. At this point, they are just associated with it, right? You have no other information whether that particular mutation really causes that disease or not. So how would you test for this, right? So given a particular plot like this is again the Manhattan plot, you want to highlight or you want to figure out, okay, that these mutations are associated, but which of them are likely causal? Which means that if I do that particular mutation, I will get that particular phenotype, right? So this is associative. I want to reach to a point where I can claim that this is causal. How would you figure that out, whether it's causal or not? Any ideas how to test causality? So yeah. Sorry? Okay, so you could do CRISPR, for example. Sure, you could do that. That's good. Yes. Any other ways you can test it? Okay. Yeah, so that would be sort of a way to rule out whether you could take a totally different population and see if they lack this variant, do they also lack that particular phenotype, right? So the second is sort of the computational way of doing it. The first is sort of the perturbation way of doing it, right? So in terms of, generally if you have to test causality, you have to do some sort of perturbation. And one of the ways to perturb is you go there, you do what Amber said, you go to that particular locus, right? Now we have techniques, right? So if you go back to the first lecture, like we are coming towards where now we can synthesize all the information that we learned in the course to try to like attack real life applications, right? At least that's how I think about the course, but you might differ, right? Based on what your experience has been in the course. But if you go back and like try to match what I promised in lecture 1, I think we are delivering everything that I promised. So I was doing this survey yesterday and I think we are very, very close to whatever I promised, right? So hopefully you'll share that feeling in, maybe not in this class, but next class when you do the assignment. But we have all the tools to sort of go and test for causality, right? So we can go here, we figure out that these are there is a particular causal variant. I can go and do a mutation here and then measure the phenotype, right? So the way to do this is to look for what happens if I perform this particular mutation. So if you perform this particular mutation, what will happen is the expression of these genes will change, right? So you can ask questions about if I particularly mutate this particular SNP, or if I go and introduce this SNP in my DNA, then what are the epigenomic effect of these variants? You can measure this in form of what Mayurakshi was saying, right? So I can have, in one case, I have a genotype which is AA, right? So that genotype refers to your maternal and paternal haplotype. So we don't get to the genetics of it, right? But we have 2 copies of the DNA, right? So one of them there is a scenario in which both of your copies have A. There's a scenario in which you have one copy from your mother, which is A, and one copy from your father, which is G. And there's a third scenario where again both of your copies are GG. Right? So we can measure based on the genotype that we identified, what is the corresponding expression of the nearby genes that are associated with this particular genotype. When we go and actually perturb, what we also end up learning is if I do a perturbation here, which genes are changing. In other words, if I mutate here, is only gene A changing? Is gene B also changing? Gene C also changing? And that is the way to capture the causality, that what is through this particular mutation, what is the nearby locuses in which things change, and how does that get associated with a particular disease, right? So if you were to go to a particular patient and profile their gene expression and also profile their genotype, you would be able to capture information like this. Maybe you will now also have this information that the one who had a genotype like AA was diabetic and this is not diabetic. So you have now established causality, because once you mutate this particular SNP, you know that genes A's expression changes exactly in this fashion, which means that if I mutate or if I introduce a particular AA mutation in this particular locus, the gene A's expression is low. It is also low in diabetic patients. And if I go and mutate and if I get a GG, then the expression of gene A is higher and it's also higher in non-diabetic patients, or vice versa. And that helps you establish causality from a disease point of view of what is causing your disease, right? So in terms of these SNPs which affect the gene expression, these are called EQTLs, expression quantitative trait loci, right? So this is slightly mouthful of a term. I forgot to write full form here. I'll fix the slide. But these stands for expression quantitative trait loci, right? So this is, it's a locus, it's a loci, it's quantitative trait because it's changing the trait, and that trait in this case is expression. So it's changing the gene expression. So this particular SNP becomes an EQTL. That's what EQTL tries to do. Once you do this kind of experiment, you can then identify if it's perturbing, for example, if it is related to gene, if this particular mutation is changing both gene A, gene B and gene C, you can find out what is the function that these genes are associated with. So in a way, you get information about what is your pathway that a particular mutation is going to get associated with. For example, in diabetic patients, it might be associated with some sort of pathway which is associated with how glucose is metabolized, for example, or how insulin is synthesized, in other words, right? So I'm making up theoretical examples, but that's what, for example, doing a mutation analysis would tell you in terms of what gene is associated with it. Okay, then comes this idea, we're almost towards the end. We can, as I mentioned previously, generally these diseases are not so easy to study, right? You do a particular, like very simple diseases like diabetes, which we know a lot about, they're not caused by one particular gene. There are like multiple different genes that are associated with it. So how do you score them? How do you score, or how do you develop, given a particular individual, if you have to score them based on how predisposed they are to developing diabetes? We have this something called the polygenic risk score, right? So this polygenic risk score essentially is a catalog of these SNPs being generated coming from different genes and their association with a particular phenotype. Again, could be anything, but let's say diabetes. What is the individual score for each of these individual SNPs, and how does overall, based on the different SNPs that can exist in different genes, what is the risk score for each individual, right? So if you have done a particular study, you have identified like 5,000 different SNPs, and you have to sort them in the order so that you can assign one particular score to an individual. The PRS, or the polygenic risk score, allows you to do that, right? So the idea is again very, very simple. You calculate a GWAS summary statistic. This GWAS summary statistic is something which you get from, collecting the patients, figuring out what their genotype is, and then you know what their particular SNP profile is. The second step is that genotype data, which are, so this you will do for a particular cohort, which shows you a particular contrast in terms of being diabetic versus non-diabetic, right? So this is done in one population where you have control over whether those people who were sort of recruited, were they diabetic or non-diabetic, right? So you will be able to assign a summary to each of the SNP, whether a particular A leads to is associated with being diabetic or not. The second thing that you do is you get new patients. For example, somebody did a GWAS on type 2 diabetes patients. Now I want to find out what is my risk profile. So I will give them my genotype data. I will give them my 23andMe data. And what they will do is they will catalog based on the scores that they calculated for each of this SNP previously in terms of their association, and calculate a combined score combining all the different SNPs and rank me based on that particular score. That what is my individual score of developing diabetes. That's basically what is polygenic risk score. And finally, you can perform a linear regression, which once you calculate your PRS score, so the PRS is just a measure of the total SNP profile that you have. If you do this over multiple different individuals, you will get a distribution. The extremes indicate that you have a tendency to develop a particular kind of a trait. Again, could be any disease, could be any particular kind of trait. This is what is being used, for example, to test, or this is what 23andMe and me also tries to do at the back end. It tells you, for example, one of the reports that it told me was what is my predisposition to developing, let's say, prostate cancer, right? So I'm a male, so I have some predisposition to developing a prostate cancer. It does this by calculating this kind of polygenic risk score. Is the general idea clear of how these risk scores are calculated? So rather than one SNP, you're taking into account multiple different SNPs, and you're essentially scoring each individual given on their genetic profile, what is their likelihood of being in the extremities of developing something, whatever that phenotype is. Okay? Okay. So what is the dark side and the bright side, right? So the association studies are a good starting point in the sense that they give you idea about what predisposition or what association you get with a particular kind of mutation. So you can use it to discover novel biological mechanisms. You can do it to discover rare variants. So rare variants, for example, are those variants which you will only see in very, very small number of individuals. But if you happen to sort of do a targeted study, for example, with people who develop a particular kind of, let's say, macular degeneration, which is, let's say, your eyes are, your power of your eyes is, or your retina is losing its power in a way. You can use this to discover like what is the possible trait, what is the possible mutation associated with those kind of diseases, which are not very, very common. Macular degeneration is not the best example, but a lot of diseases happen because a lot of rare diseases happen in India because any guesses why do we? So India has a number of rare diseases that happen. Any ideas why that happens? We haven't covered that actually in the class, but you can take a guess based on what you know about India and how marriages happen here. Yeah, so there is a lot of endogamy that happens in India, right? So people marry within, again, this is slightly like every time I talk about it, like somebody comes up and says it's a very uncomfortable topic, but it's also a very reality that people marry in their caste or in their religion and so on, right? And because of this, what happens is you end up developing, like you have very, very similar traits. So if you had very, very similar ancestors, and because of whatever goes wrong, if something went wrong with one of your ancestors, that keeps replicating, and if both of you are coming from the same ancestor, you end up marrying and your offspring has much higher probability of having two copies of the same wrong sequence that basically causes a number of diseases, right? So a number of rare diseases happen in India because of that reason, that there's a lot of endogamy, like people marry within their families, people marry within their religion or within their caste. So that causes a lot of problems overall. So you can use this GWAS to study, right? So if I can go and, for example, Ashkenazi Jews are also another population which marry within their community a lot. So there is a lot of endogamy that happens there, and they are also known to cause a lot of these rare variant diseases. So if I go study Ashkenazi Jews, I can figure out what exactly is going wrong, which is that particular mutation that is getting copied again and again, which is also associated with a number of diseases, right? So there are studies that are happening in India also, which trying to understand like what is this which are these rare variants, because most of the time these get undiagnosed, right? Because if you have a rare disease, well, good luck, because we don't know why it happens. We have no way to cure it. So if you want to cure it, you have to figure out why it happened, and that can be done using these studies. So you cannot use it again, like, so this is the dark side, which is it doesn't tell you about whether it cannot directly be used for prediction. So it is again an association, so I cannot directly use this data to tell me whether I am going to have a disease or not. It will always remain a prediction, and that to a very, very loose prediction. It is just going to be an association, because I have no causality angle to draw unless I go in the lab and do an experiment to tell you that, okay, this variant for sure, if I mutate it, it causes things. Also, biology is much more complicated. One mutation often will not do anything. One mutation and a collection of mutation, which is basically what broadly is referred to epistasis, is if two mutations happen within certain particular boundary, then their effect can be much larger as compared to just one single effect, and that's what happens a lot of times. GWAS is not the way to capture it, right? Because it treats each mutation as an individual mutation, so it doesn't take into account the interaction effect that some of these mutations can have. And then you have to do more experiments to figure out whether there is causal variance or not. Okay, so I'll end it here, and for the first time in history, I think we are ending before time. If you have any questions, so very few of you have unfortunately submitted the assignments, and I think the other problem is who have not submitted are probably also not here, so there's no point like figuring out what is going wrong. But if anybody who is here who is not submitted, if you can just tell me what went wrong or what is not working, I'm happy to help. Yeah. So I put the HTML also, which is knitted using the same framework. So if it is not working because of the PySRDB stuff, you can remove it. It's not I think I removed it anyway. It's not required. But it will, if you try to knit it, it will also tell you, it will give you an error of why it is facing an error. So if you show me that error, I can tell you, right? So the one principle, like for anybody to help you, is like you have to show what you did, right? So that's when I can give you feedback of what did not work. So otherwise, if it is not working, then it cannot be working for multiple reasons. So if I need to know the specific reason what did not work, so then I can tell you what would happen. So the knitting takes time because I think you're trying to do, you're trying to do a lot of things in that one thing, right? So that is the reason it tries to do a lot of. That's why it's a bit slow. We can do it right now actually if you want. What errors? Like what? So let's do it actually. Let's do it right now. We'll see what it looks like. Okay, so I'm going to